In this video, we'll be discussing about the human herpes virus 4, that is Epstein-Barr virus. As it is a member of Herpes viridae family, it will be enveloped, linear and has double-stranded DNA. Epstein-Barr virus is also known as kissing disease. Therefore, its transmission will be via kissing, close contact, sexual intercourse, or can be due to blood transfusion and bone marrow transplant. Now its pathogenesis. Most of the virus, when enter our body, goes to T cell first and then to B cell. But this Epstein-Barr virus goes directly to B cell to attack our B cell and causes polyclonal cell activation. And the receptor through which this virus enters the B cell is CD21-CR2. Now, what kind of molecules do Epstein-Barr virus has? So it has LMP1, EBNA2 and viral interleukin 10. This viral interleukin 10 is anti-inflammatory. That means the virus will itself stop the inflammation, and that's not good for us. This LMP1 acts on CD40 pathway and EBNA2 activates the SRC proto-oncogene. Now about the clinical features, we know that it is called kissing disease, but it is also called glandular fever. And its incubation period is 4 to 8 weeks. Easy to remember because human herpes virus 4. And this is 4 into 2. Simple. It causes infectious mononucleosis. And as the name is kissing disease, the clinical features will be sore throat, lymphadenopathy, and the patient will have fever. And also, the other name is glandular fever. That means glands will be affected, like salivary gland, spleen. So the clinical features will be splenomegaly, hepatitis, encephalitis, fever, sore throat, lymph node involvement like lymphadenopathy, and the less common ones are arthralgia and lethargy. Now, other than infectious mononucleosis, it also causes malignancy. Like under carcinoma, we have 2, lymphoma, we have 2, sarcoma, we have just 1. Starting with carcinoma, we have gastric carcinoma and nasopharyngeal carcinoma. Gastric carcinoma is most common. Under lymphoma, it can cause Hodgkin and non-Hodgkin lymphoma. And under sarcoma, it can cause leiomyosarcoma. Moving towards lab diagnosis, when we take blood sample, we find something like this. If you notice carefully, it is like a ballerina skirt, like a girl in ballerina skirt. These are atypical lymphocytes, also called Downey cells. And this cell is T cell, not B cell, because B cells are already attacked by the virus. The next test will be heterophile antibody detection. In this, we have 2 tests, Paul bundle test and monospot test. This Paul bundle test is antibodies react with sheep RBC, as it is heterophile, means 2 different species, and the other is monospot test. This is antibodies react with horse RBC. This was all non-specific, but now we have specific antibody test. And the Epstein-Barr virus has capsid, so we have test for that, the antibody to viral capsid antigen test. The other test is antibody to early antigen. This is for early detection And what else do this virus has? It has EBNA2. So we have test for that. Antibody to EBNA2. And the most sensitive and specific test will be nucleic acid hybridization test. And for treatment, we will give acyclovir for oral hairy cell leukoplakia and rituximab because it is anti-CD20. Thanks for watching. Don't forget to like and subscribe and share with your friends.